Thank you, Manuela, for the introduction. For those of you that don't know me, I'm Babis. Well, actually, most of the parts that I usually cover in my presentation have already been covered by Anastasia and Petros, but I would say that we can summarize actually stuff regarding common practices, equipment, and that we usually utilize in our laboratories at the IMBB. So today we're going to be talking about the different biosafety levels that the laboratory actually can belong to, depending on the reagents that it deals with. I'm going to remind you about the safety practices that you have to follow in our laboratories. I'm going to give you a couple of recipes and ways to decontaminate either areas or equipment. And at the end, I'm going to talk again about how to contain or what to do in case of a small or either a large spill within the laboratory. Now, regarding the definitions, when we talk about biosafety, we usually refer to three things, and I'm going to actually follow these three things when I'm going to be talking about the biosafety levels 1 and 2 that all of our laboratories at the IMBB belong to. So from the beginning, I have to tell you that all IMBB laboratories are biosafety level 2. You're going to see later what I'm talking about, this biosafety level. But as I said, there are three things actually that we pay attention when we refer to the biosafety levels of the different laboratories. So one of them is are the laboratory practices that we have to follow. The second are the equipment, the laboratory facilities that we need to utilize depending on the reagents we're working with. And the third is the safety equipment that we should have in our laboratories. And we need all these. Okay, we need biosafety since if we're working with infectious microorganisms that are potentially actually dangerous or even not dangerous to people or the environment. And actually, we do need biosafety practices because we need to protect. We need to protect ourselves. We need to protect everyone that is working in the lab, including for example, students. We have a lot of either undergraduate students or master's degree or PhD students. So all of these have to be protected following specific biosafety practices. We also have to protect co-workers or visitors. Remember that we have this visiting program at fourth. So we have even a lot of high school students visiting our research laboratories. So there are certain measures that have to be taken even if we have visitors. We need also to protect the lab support personnel. Imagine the ladies that dispose the garbage, that have no clue about the reagents, the potentially biohazardous reagents that we work with in our laboratories. So we have to protect the ladies and not to get infected, let's say, with such a reagent. Quite importantly, and many people actually don't pay attention to that, we need actually biosafety practices to protect either the products or, more importantly, for basic research laboratories, the experimental results. So if you have a contamination, of course, this is gonna affect even your experimental results. So as I said, certain measures have to be taken also to protect the environment. And here Petros talked a lot about that. So we need certain practices to protect the environment, but also our laboratory or a classroom if we're talking about students. So a biohazard is any microorganism, toxin, allergen, in general, any agent of biological origin that could potentially cause disease to humans. And here we refer to infectious agents or pathogens such as, for example, bacteria, viruses, parasites, fungi. We do actually culture a lot in our laboratories. We're talking about human tissues. Sometimes one might perform actually such experiments, especially the laboratories that collaborate with IMBB and are located over at the medical school. Infectious agents actually could also include primate tissues, non-human, for example, or even animals. We do work a lot with mice, actually. Biological toxins are also biohazardous materials or reagents, and we refer here to botulinum or tetrodotoxin or ritin. I've never heard the lab working with these agents, but you never know. We do use tones of recombinant DNA and RNAi. So everyone is working, I guess, most of the labs are working with plasmids, linear naked DNA, or even synthetic oligonucleotides. And imagine that all these are, uh, biomolecules. So these could potentially be integrated in the human genome, especially when they're combined with viral vectors such as adenoviruses, murine leukemia virus, or lentiviruses that we utilize in order to transiently transfect cells, either human cells or others. So all these are actually potential biohazardous materials, and for all these we have to follow certain measures to protect ourselves and the laboratories. The equipment that could potentially be hazardous to ourselves are needles, centrifuges, and water baths. Water baths because actually in a certain temperature the microorganisms could grow up. Centrifuges because, for example, a tube could break and the contained material could be spilled over and create aerosols and potentially infect the people in the lab, but also needles that we utilize in order to work with the biohazardous materials. If we accidentally inoculate ourselves, then this could potentially be hazardous to us. I remember myself injecting ethidium bromide in my bloodstream. Mm, Not a huge quantity, but it could potentially be dangerous. So this equipment, the needles, the centrifuges, and the water baths, whenever we need it, we use it. And we work with the biohazardous reagent. We have to really be quite careful, actually, not to create an accident. The routes of entry of these biohazardous materials in the human body could initially be the skin, okay? So that's why you have to wear gloves or lab coats. Quite importantly, we have to wear closed to shoes because either chemicals or either biological reagents could be spilled over. We definitely, especially the ladies, should not wear makeup because that's where biological reagents could stack. The eyes are actually a quite easy way of entry of biohazardous materials. That's why we need the biosafety glasses. Biohazardous materials could be ingested in the digestive tract. That's why we never eat, drink, or smoke in the lab, and food is not allowed in the lab. Injection into the bloodstream, as I said before, that's why we need to properly dispose all the sharps in certain containers, or a biohazardous material due to aerosols, for example, could be inhaled. And here, certain masks actually could be protective. We need the safety practices before performing an experiment. For example, when we collect or prepare a sample for transport, imagine if we are talking about, let's say, blood samples that have to be prepared and transported from the, let's say, the hospital to a research laboratory in order for certain exams or experiments to be performed. So the safety practices could be applied before the experiment, during the experiment, but also after the experiment, where we have to dispose all the contaminated material. Um, okay. Remember I talked about the different biosafety levels, and I said that all Imbb laboratories actually belong to biosafety level 2. So the designation either BL or BSL actually could be found outside the door of a lab. We don't actually have it here at the Imbb, but you could actually see such signs in certain laboratories designating the biosafety level and therefore the precautions that have to be taken before entering in such a lab. So biosafety level 1 laboratories actually don't deal with that dangerous, let's say, material. Usually they deal with agents that are not known actually to cause disease to humans. They are of minimum hazard, and these include certain bacterial strains such as DH5 alpha or E. coli strains, and also Saccharomyces cerevisiae. So BL1 laboratories would not deal with potentially hazardous microorganisms or materials. In any case, the special practices that we should follow actually include mechanical pipettors and not mouth pipettors. Now you would say, I've never seen anyone actually pipetting by mouth. I did see when I was a student, actually people pipetting by mouth, either radioactive material or even other reagents. So in any case, whenever we pipette, we should use mechanical pipettors. As I said, never drink, eat, or smoke in the laboratory. Quite importantly, never store food in the refrigerators within a laboratory space. There are designated refrigerators outside the laboratory space where one can actually keep their food. We should wash our hands frequently. When I was a student, I would do that 10, 12, 15 times per day. Every time I was completing an experiment, I would wash my hands. And actually, this is quite important. Such a practice actually is quite important to protect yourself. We should minimize splashes and aerosols when we work with biohazardous material. I'm going to show you later how. We should decontaminate the work surface daily. I would say even not once, but many times within a day, especially after certain experiments, we should decontaminate our bench. Waste should be handled properly. And we should have, for example, an insectarium and a mouse room, and we do have these in at the Imbb in order to breed, let's say, insects or mice. Now, regarding the personnel protective equipment, as it was mentioned earlier by Anastasia and Petrus, we do need to wear lab coats to protect ourselves actually from being contaminated by biohazardous materials, safety glasses, and gloves. Depending on the work we do, we should wear certain kinds of glasses, as Petrus actually elaborated earlier. Now, for biosafety level 2 laboratories, here we're talking about reagents, biohazardous materials that could potentially be dangerous to either ourselves or the environment. And here we refer, for example, to human blood or body fluids, to certain E. coli strains, to Clostridium botulinum, retroviral vectors. We do use a lot actually in order, as I said, to transiently transfect, for example, cells or to express certain DNA fragments, and human cells in cell culture. And we do culture a lot of cells. Now, regarding the laboratory facilities, you already heard about that. We should have adequate illumination within our laboratories, and quite importantly, eye wash facilities, and you will find them actually in the corridors outside of the research laboratories. We do have negative air pressure, especially applied either in the fume hoods or the biosafety cabinets within tissue culture rooms. And of course, for disposing biohazardous materials, we do have autoclaves available where we autoclave all the potential contaminated equipment plus any material that before released to the environment. So everything is autoclaved in order to exterminate all microorganisms. And of course, we do have biological safety cabinets. As I said, you're gonna find them in the tissue culture rooms, specialized rooms actually, where we grow either human or other species cell lines. So there are, as I said, specially designated such spaces in order to perform experiments with tissue culture. So the work practices that we have actually to incorporate in BL2 laboratories are the same as the BL1 laboratories, okay? Mechanical pipetters, as I said, washing our hands, minimize splashes and aerosols, decontaminate the workspace, special waste handling, and insectarium and mouse rooms. Quite importantly, and we did have questions from Manuela earlier, actually, we should provide special containers to dispose the sharps. These containers should be actually specially marked. They are puncture resistant, quite important, and they are leak proof. And they should actually be disposed, as you can see here, do not fill above this level. They should be disposed before actually the sharps reach the top of the container. There is no way that you're going to put sharps such as needles or even glass slides or any other sharp in the normal office waste containers. This could potentially be dangerous, as I said, to lab supportive personnel. If something breaks, let's say if you have glassware and it contains a biohazardous material and it breaks within the lab space, never touch the glass with your hands, even if you wear glasses. Actually, it's better to use cartons in order to dispose the contaminated material first to a bag and then put it in proper waste containers for biohazardous materials. Something quite important. I think almost everyone is doing this kind of mistake. We do wear lab coats in order to protect ourselves. And I mean protect ourselves because the biohazardous material or agent will be spilled on the lab coat. So when you leave the lab area, you should always leave your lab coats within the lab. Otherwise, you move around with a biohazardous material, and this potentially could contaminate other areas within the institute. So when we exit the lab, we should take out our gloves and also the lab coats. And quite importantly, we should never bring animals in the laboratories. I know that a lot of labs are doing this for a few hours before performing an experiment, but definitely we don't breed or maintain for longer periods of time animals within our laboratories. Now, apart from the practices we should follow, the personnel equipment we should have in a BL2 laboratory, as the BL1 includes the lab coat, the safety glasses, the gloves, and on top of biosafety cabinets, as I said, in the tissue culture rooms. Now, BL3 laboratories, we definitely do not have a BL3 designated research laboratory at the Imbb. I'm not aware if there are such laboratories over at the medical school, since they are potentially dealing with quite biohazardous microorganisms from human samples. So BL3 laboratories are designated for dealing with, agents that they are potentially airborne transmitted, and they could actually cause serious health problems. And here we are talking about microorganisms such as Mycobacterium tuberculosis or Coxiella burnetii. And also BL4 designated laboratories. Again, here we talk about quite dangerous agents that could definitely be lethal to humans, and these include, for example, Marburg and Ebola viruses. So these are the 4 biosafety levels of laboratories that deal with biohazardous materials. As I said, Imbb laboratories have clearance for working with BL2 designated biohazardous materials and microorganisms. Now, More specifically regarding the safety practices, simple things you can do in order to protect yourself, others, and the environment when you work with biohazardous materials. So as we earlier said, okay, it goes without saying, wear gloves, depending on the experiment, and most of them actually do need you to wear gloves. Wear safety glasses, especially ladies. Well, most men, unlike me, have also long hair, especially ladies have long hair. You should keep them on the back, okay? Keep hair on the back so they are not sweeping around and being contaminated with microorganisms. As I said, no smoking. I even quit smoking. No eating or drinking or even storing food in the lab, and wear closed-toed shoes, okay? Otherwise, mostly, I had this accident in the past, mostly with chemicals, actually. Not my feet, but clothes. I destroyed a lot of clothes when I was not wearing a lab coat. As I said, washing hands during the day, okay? It's actually been the best way of not being contaminated. And as I mentioned before, do not wear the gloves and the coat outside the laboratory where you perform your experiment. Now, in order to protect yourself, it would be better actually to use plasticware instead of glassware, since if the glassware breaks, then you have to follow a certain procedure in order to dispose the potentially contaminated equipment, but also area. Earlier, I said that you can avoid actually creating aerosols or spills within the lab. And you could do that if you pipette, let's say, potential biohazardous material on the side of the tube instead of going directly on top of the tube and pipetting in the middle of such a tube. So if you pipette gently on the side, then this decreases the risk of creating aerosols with potentially biohazardous material. When we work in biosafety cabinets, we could actually use a container where we put all the contaminated, let's say, tips within the biosafety cabinet instead of every time getting out of the cabinet and disposing such materials. So having such a container dirty, I would say dirty because you put dirty stuff, potentially contaminated stuff in the biosafety cabinet could actually save you time, but also it could decrease the risk of contaminating the surrounding areas. A few more things when we work, Petros, I think, mentioned a lot, especially for the fume hoods, but I'm gonna say actually a few more things about the biosafety cabinets. As Petros said, because there are actually filters and special waves of air within the biosafety cabinet, the sash should be pulled down actually, and having a minimal space so that we can put in either equipment or our hands. We should never block the grill, especially because this is where the air actually is getting out, is going to the top, and then it's being pulled out from filters and motors on the top. So never put stuff on the grill. If you don't want to contaminate your material, but also contaminate the outside area, then your material should be put on the back of the cabinet. And since the only movement here is by ourselves, then our hands should be moving slowly so that we don't cause turbulence and increasing actually the chance of biohazardous material to get out of the biosafety cabinet. When we enter the biosafety cabinet, we should go straight and we should avoid actually swiping our hands or our clothes on the grill. As I said, never put material on the grill. And it could be good actually to use a container where we dispose contaminated, let's say, tips or other material. Quite importantly, before we work in the biosafety cabinet, we decontaminate everything. after we're done with our work, we decontaminate everything within the biosafety cabinet, but also the material that we're going to be taking out, such as culture media, for example, before storing them outside the biosafety cabinet. So we decontaminate in the beginning, we decontaminate the area and the materials we use after we're done with a certain experiment. Now, the best way to organize your work in a biosafety cabinet is to maintain 3 discrete areas. One would be the clean area, where you do have all the materials actually that were not opened, so there is no potential contamination. You can have a contaminated area where you also have the container of the contaminated, let's say, equipment such as tips, and then you have your working area. So the contaminated and the clean area are actually quite discrete areas within the biosafety cabinet in order to minimize the potential of cross-contamination or taking out materials from the biosafety cabinet that could potentially contaminate equipment and people. Petros actually did talk a lot about these. Fume hoods that we have in our labs are to store flammable material and also acids or bases. Okay, these are different from the biosafety cabinets that we have in the tissue culture rooms. So you have to be careful what to store in each hood, in each cabinet. Okay, flammable materials have nothing to do and should never be stored in biosafety cabinets. The designated fume hoods are within our labs, and they do have pumps actually taking out all the fumes. So we have to be careful because certain accidents can happen if we don't properly store in the designated hoods the material we have, such as flammable material. Quite important, and actually you're not going to be responsible, but the personnel, the technicians that work in the tissue culture rooms. But if there is not such person, be careful that be careful to use filters that separate, let's say, the flask, which will contain the contaminated media from the vacuum lines. So we don't contaminate with whatever biohazardous material all the vacuum lines in the building. So such a filter actually could be beneficial to separate, okay, the permanent, let's say, facilities in the material from the biohazardous material that we're going to be collecting in such waste containers, such as glass or plastic flasks. So these filters are beneficial actually to separate the different areas. We talked about that earlier. Sharps should be contained in approved sharp containers. As I said, we should never overfill them. And after they are packed with sharps, then they should be closed and put in specific biohazard boxes before being disposed. Now, um, what we can use in order to decontaminate, or what are the different methods that one could use in order to decontaminate or kill, let's say, potential biohazardous microorganisms. There are 3 ways actually to decontaminate. One is the sterilization. This is great because not only destroys microbes, but also destroys the spores that can potentially be created. Disinfection is great, but it can destroy most microbes, but not necessarily the spores. And antiseptics are good if we want to stop, let's say, microorganisms growing, but it does not necessarily mean that the microorganisms are destroyed. So the methods that are applied for these different ways of decontamination are heat. Okay, we do use heat in the autoclaves, so that's the way we use when we dispose stuff from the tissue culture rooms, for example. because steam heat and the increased pressure will destroy even spores if these spores exist, let's say, in contaminated equipment or disposed equipment. Chemicals such as bleach, ethanol, hydrogen peroxide, ethylene oxide, or paraformaldehyde could be used in order to kill microorganisms and also radiation. I remember there was a container actually where we would put our pipettes in order to provide radiation and kill all microorganisms that potentially had contaminated our pipettes. And I think we do have that in at the Imbb. But quite importantly, no matter what decontamination practices we utilize, it's actually quite important to follow certain microbiological practices, as we mentioned earlier, such as washing our hands, wear gloves, lab coats, and certain practices we should follow in order to protect ourselves and others. Now, regarding the actions we should take if we have a spill, if it's a small spill, for example, then what you can do is that you can soak paper towels in 10% bleach, put it on the spill or wipe out the spill, then repeat this. Then replace the protective equipment such as gloves, and then wash your hands, and it would probably be fine. But if the spill is larger, then you should do about the same thing but for a longer period of time. So again, you can soak paper towels in bleach, put it on the spill, leave it for at least 30 minutes, because even for bleach, it takes time actually to kill potential biohazardous microorganisms. Then dispose the paper towels in designated containers, replace the protective equipment, inform your colleagues. Instead of going around and screaming, you could just inform your colleagues. Depending on the spill, you could ask them to get out, to leave the lab for a certain period of time, at least 20 to 30 minutes, closing the door, open the windows, um, and let them come back after you are done with decontaminating the area. Definitely a safety officer, and you've seen from Petros the phone numbers of all the people responsible for biosafety at the Imbb and forth. So the biosafety officer should be informed, and definitely the principal investigator, the person responsible in the lab. Now, how you can prepare by yourself different disinfectants. As I said, you can prepare, for example, 10% bleach. This should be prepared daily, actually. Otherwise, salts are being created by reacting with oxygen from the air, and then it's not actually worth using it. So daily prepare 10% bleach and use it in order to decontaminate areas. Or even better, you can use 70% isopropyl alcohol or ethyl alcohol in order to decontaminate areas. Most people actually, I guess, don't know that, but 100% ethanol is not good because, organic solvents are not great for dissolving biological materials such as microorganisms, such as bacteria. What you need here is a certain percentage, a certain fraction of water, because most biohazardous materials such as microorganisms are being dissolved in water. So you need 30% water to dissolve the microorganism, and then you need the 70% ethanol in order to to dissolve, for example, membranes of the microorganism and kill it. So 70% ethanol would be a great disinfectant, and what we usually use in order to disinfect our benches. If you want to be sure, 10% bleach is great, but it leaves a residue, so you have to clean it up with water. Now, if you want to prepare by yourself a disinfectant that would be actually capable of destroying SARS-CoV-2, then the World Health Organization actually has the recipe available, and we do have all these chemicals in our laboratories. Instead of 70% ethanol that I said before, here you need 80% alcohol, 1.45% glycerol. This is not of any use to our bench, but it's of use to our hands. To maintain the softness and not leaving the hydrogen peroxide actually making our hands dry. So 80% alcohol, 0.125% hydrogen peroxide. This is the recipe actually for disinfecting SARS-CoV-2. If you want to use it on your hands, then 1.45% glycerol would make the difference. So that's all I wanted to discuss with you about. I would be happy to take any questions you may have.
I don't see any questions coming up at the chat. So
Manuela, can I ask something? I'm curious. Sorry, Bobby.
No.
The coronavirus, if you do work with coronavirus, what biosafety level do you need?
There is only one lab working with coronavirus, and I think that's what's his name? The one who's performing
Dr. Sorvino.
Oh yeah, curious, Mr. Sorvino.
I mean, if you if you do the test, this test that we do, it's okay to do them here. You don't send them outside.
Sure, definitely. Now, if you work with coronavirus, it is my opinion that it would work at least in a BL3 laboratory. If you work with live virus, because it is known to be airborne transmitted. So that's actually making the difference going from a BL2 to BL3. If it's airborne transmitted, then you should definitely work in a BL3 laboratory. So I would say BL3 laboratory, and we definitely don't have such laboratories at the Imbb unless you don't work with a living virus, but just parts of the virus, let's say a protein. Then I would guess a BL2 would be fine. And we do have labs working with such material, BL2 designated and not BL3.
Okay, thanks.
Actually it's the most dangerous thing for creating a vaccine the traditional way, meaning to produce SARS-CoV-2 in large quantities, then kill it and then use it in a vaccine, because you need such laboratories to contain the living virus and not let it spread to either the personnel or outside, let's say, the factory that creates the vaccine. So that's the most difficult thing, actually, to maintain such airborne transmitted microbes.
Good excuse for AstraZeneca. I don't have such areas. I don't produce vaccines. Bye-bye. All right. Thank you, Bobby.
Sostos, Pedro. Manuela.
A question from Manuela.
Yeah, I see it. How do we dispose of vials and pipettes that contain human blood? They should go actually to the containers that are found in the tissue culture room, because these actually, I think they have red or blue bags. I don't actually remember. Blue, are these blue?
Blue are the bags of
okay. Yeah, in the US they were red. Here they are blue, because these will be actually be autoclaved before being disposed to the garbage. So they should definitely be disposed in the tissue culture rooms in the designated containers, which have blue bags, and they're next to the biosafety cabinets for Manuela. Manuela Linardaki, yeah, okay. What are we doing with the rest of the material be used for the COVID test? ER sticks in case they are positive. I would say the same thing. If they go to the same blue bags, they will be autoclaved. as soon as no one is touching inside the bags and the containers, I guess. But that's what I would do, because they're going to be autoclaved, and I think they're being collected and autoclaved in almost a daily basis. Then I would dispose them there and not in the normal garbage.
Okay, if there is no other questions, we thank Babis very much for his presentation. And we continue with the last part of our seminar series, presented by Mr. Kostas Kourouniotis, and deals with safety issues with handling living animals.
Okay.
Hello, Kostas.
Hello. Just a minute. I'm trying to. Oh. Okay. Oh, just a minute. I'm not very.
It's tricky.
Yes. Wait a minute. Can you see the? Have I done the sharing? No.
Not yet.
The green.
You push the green button, the share screen, and then you select which window you want to share.
The thing and oh. You covered the whole όλη την οθόνη και προσπαθώ τώρα να μπω.
Κώστα, αν θες, δεν χρειάζεται να καλύψω όλη την οθόνη. Μην κάνεις present.
Ναι,
άνοιξε το PowerPoint και κάνε το share. Έτσι κάνω εγώ μάθημα.
Ναι, το θέμα είναι το πάτησα τώρα και μου γέμισε την οθόνη. Αλλά...
Πάτα escape να φύγει από όλη την οθόνη.
Και άλλους προβολείς, ας πούμε.
Ωραία. Και τώρα μεγάλωσε το όσο θέλεις και μιλάς.
Οκ. Λοιπόν, κάνω share screen.
Είναι ήδη shared. Αν έχεις πατήσει μία φορά share screen, είναι ήδη shared. Πάτα escape να μην πηγαίνεις όλη την οθόνη. Μεγάλωσε το όσο θες και μιλάς. Προς το παρόν δεν βλέπουμε τίποτα.
Τώρα, εντάξει, μια χαρά.
Ωραία. Μεγαλώνω από πού?
Το βλέπουμε.
Εντάξει.
Ναι.
Οκ. Λοιπόν, κάτι να πάω στην αρχή. Hello, I'm Kostas Kourouniotis. I'm the veterinarian from the Imbb Animal Facility. Today, we'll say some things about the safety issues in experiments with living organisms. It will be a short presentation because most of the things I could say have already been said by the previous speakers. Mostly we talk about the animal facility and mice because that's the organism we have in the animal facility. Now, for those who come for the first time in the premises of the animal facility, please ask to be informed about the protocol of entry and the general behavior in the animal facility. For that, you can contact us, the phone numbers. For one thing, in order to enter in the animal facility, you have to be covered from head to toe with coveralls, surgical cups, shoe covers, gloves, etc. Before you enter in the animal rooms, you have to sanitize your hand and wear gloves. Now, when you enter, take some time to look around and find out where the emergency exits are. Please remember where the exit is, if there are any emergency exits, where the water faucets are so you can wash after a splash or something, you get contaminated, where the fire extinguisher is, and where the telephones are, in case you have to contact with the outside world. The main gate, you can use it if you get locked in. It has happened in the past. Technical service if there was a leak in water pipe, electrical failures, etc. And if you want to contact the other workers in the animal facility, you use the numbers to see for the animal facility. Now, for some more, for some more general issues, that applies so in the animal facility and in the laboratories in general. Always wear gloves. Of course, don't eat, drink, or smoke in the animal facility. Try and keep your workbench clean and tidy. That will help you. It will help you, so you won't have to, let's say, chase a mice between little bottles and boxes if it escapes. Organize and prepare everything you need before you start. So again, you won't have to look for tubes, syringes, scissors while you hold the mice. Always have a second empty cage near you when you work with mice. So you take the mouse out of the cage where they kept in, do whatever you have to do, injections or whatever, and then put it on the empty cage before you take the next mouse. Never leave it on the top of the cage because it will just jump off and run away. And of course, be careful with the things you use. Now, about handling mice, the most important is to be calm. If you're nervous, the animals will get nervous and it will be very difficult to work with them. Now, in order to restrain a mouse, you catch it by the tail, put it on top of the grill of the cage, then grab the mouse by the neck, just behind the ears, and try to get as much skin as you can between your thumb and index. That will keep it immobilized. But if anyway you do get bitten, please keep cool. Don't shake or yank your hand because that will break its neck, especially in younger mice. And of course, it could send it flying away, which has happened before. Now, because most of the animals we use are mutated, we have to be very, very careful not to let them escape in the environment. Any mouse that has escaped, it has to be found dead or alive. We have to be very careful with that. Now, I try to, if it works, show you how you catch and restrain a mouse. Just a short video. Okay. Oops. Yeah. Now, after you finish, before leaving, clean and disinfect your workbench. Any sharp object you needed, put it in the special containers. Any cadavers or tissues, they should put in the freezer for incineration. There is a freezer in the side entry of the building C down at the animal facility. When you finish with the mice, the dead bodies, please don't wrap it up in the aluminum foil. You can wrap it up in the paper tissue or just put it in the plastic bag, because after incineration, the aluminum turns to dust and pollutes the environment, and we can inhale. Any contaminated materials, they should be sterilized in the autoclaving. If you use the autoclave, you have to be very careful, never open it before complete decompression, and use protective gloves so you avoid accidental burns. Now, about the animals, we have we don't need anymore or we have to sacrifice. If it's a large number of animals, we use CO2, carbon dioxide. If there are just a few, we can use cervical dislocation. But before we throw them away, we have to be very, very sure that they are dead. For that, we have to wait for rigor mortis, especially when we use CO2, because they might seem dead, and after a quarter or so, they wake up again and start moving. I have found in the past live animals in plastic bags that were put in the freezer. Now, newborns or very young animals, they are very, very resistant to CO2. Actually, they just don't die. So instead, we use decapitation. It's not very pleasant, but we have to do it. We use small scissors to decapitate them. Yep. Some more general preventing measures. All those that work with animals should be vaccinated for tetanus. Few things about zoonosis. One of the most important when we work with mice, it's leptospirosis, which is transmitted by direct contact or with urine or other tissues like embryo fluids from abortions, etc., or just water contaminated by them. But you don't have to be worried because you can get it from wild or free animals. Where our animals are supposed to be pathogen-free. For that reason, we have a periodic health screening, so they're not dangerous. Well, we have a lot of zoonoses. Some of them are like rat bite fever when we work with rats, tularemia, etc. But the real problem is allergies. Statistics have shown that up to or even more than 50% of those that work for a long time in the animal facility will sometimes develop some form of allergy. Allergies come from allergens like proteins from animal hair, follicles, saliva, feces, etc. Dust from the litter, from the wood shaving. Impaired air makes things worse. If we know that we suffer from the allergies, we should report it. Most common symptoms, usually we just have sneezing, coughing, eye irritation, but we can get asthma and very rare anaphylaxis or anaphylactic shock. Sometimes it happens after a bite if saliva enters the blood circulation, but it's very rare. To protect ourselves, we can use protective masks, coveralls, goggles to protect our eyes, gloves, etc. Okay, that's all I had to say. I said it was a short presentation. If you have any questions and I can ask them, please ask. Thank you.
Thank you very much, Kostas.
Okay, thanks.
I can see no questions at the chat area. I have seen a lot of people have been sending their names and their email addresses at a time that I have not requested for that. The request for names and email addresses was for the purpose of tracking down who is participating. I will ask you to do the same right now, no matter whether you have just sent it or not, because like I said, this was a means to track down real participants. At this point, I guess the seminar series are finished. And I would like to thank again all the speakers for their presentations. and look forward to the seminars of next year, which will be approximately around the same time period in 2022. I thank all the participants again for their presence in these seminars. If anyone has any comments or any questions, we are still here for 5 minutes until everyone manages to sign up before we close. And we will say goodbye.
Okay.
So I will be here. We have the platform on for another 5 minutes. So if anyone wants to say something or ask something in the meantime. Okay, there are some questions coming, but as they come together with the names, I have a difficulty following them. So please give me a minute.
Manuela, μια ερώτηση. If we measure small animals like worms on microscope slides, where is it safe to put them after? We used to clean the slide with ethanol. Is it safe?
With worms, you mean C. elegans?
Worms, it says worms.
Okay, so you saw this on the chat?
Yes, yeah, 1234.
Yes, I've seen it. In fact, I don't know. I cannot answer that because I don't know what is the right practice for worms. I guess
the slide should definitely go to the sharps container. That's one thing, the slide. Now, how to kill C. elegans? I don't know. Is ethanol enough?
I'm not sure.
I guess they could be put in 10% bleach, in a container with 10% bleach, and then after some time, the slides, the microscope slides, could be washed out with running water and put in sharps container. That's what I would do. The same we do with bacteria, where we put them in diluted bleach.
Okay, there is another question whether the seminars are somewhere accessible to new members, to new lab members. So the answer to that is that all the presentations are stored in the site of Imbb under the description safety issues in the intranet of Imbb. So you can find the presentations there. We will try to put the updated versions there as soon as possible. So soon enough, you will be able to find even the ones that were presented today. To enter this link, though, you need the password, a username and the password. And if you actually want to access this page, you have to send me an email so that I can send you the username and the password. At least until we remove this password, which I'm not sure whether it is necessary, but anyway, it's not up to me to decide. So,
Manuela, I can, if the speakers have no objection, I can put them also on the ISL website, which is already open.
It's open access. Okay.
But sometimes you have, copyrighted material and so forth. That is an issue. So we have to get the okay from Babis and Anastasia.
No, this time I'm fine with that. But
My talk is already okay.
Yeah, I don't know if there was any special reason for the Biosafety Committee that had restricted access. I don't know if there is something else, but I mean, like personal data protection, but for me it's fine.
Okay. So
As soon as the participants are not visible to the public, probably it's okay.
it's the presentation. Usually your face will show. That's the thing there. My face is already out there, so I don't have a problem. That's why I'm asking.
Yeah, I'm fine. I'm fine.
I will ask Anastasia.
If it was a lecture, let's say at the university, I might had material protected, but now, no, I feel confident. It's fine.
Sometimes you have, the images that you take from a source, and you don't have permission to use it. But usually they are sort of flexible. Nobody has been sued for using an image in a safety presentation.
Well, I took them from the internet, so I guess I do have the right actually to use them. Since they're out there.
This is not always the case. That's what I'm saying.
I hope Angelina will not sue me.
That's okay. I think you're fine.
Okay.
Okay, I guess the 5 minutes are gone. So I will see you next time. Well, no, I hope I don't see you next time, because I hope that next year we will have videos ready, so you will not be obliged to perform these presentations anymore.
I don't mind.
Anyway,
you always need updates sometime, maybe.
Yeah, it's true, but we will try to prepare them such that they can be updated easily, the little videos. So Petros, I will come and talk about it at some point with you.
Okay. Yep.
Okay, so bye now, and
okay.
Thank you.
Bye bye.
Have a nice day.
Thank you very much. All of you. Bye.